what I'm interested in and what I'm going to talk about is a way of conceptualizing information in biology, really. And there have been many attempts at doing that, especially in the past decade, since a certain paper came out, and we'll talk about it. But what I wanted to do more specifically is try to see whether I can really quantify some sense of information that relates to biology, rather than just talk about conceptual issues and issues of meaning and analogies and certain other concepts from maybe from philosophy of science that people have been using, the philosophers of biology have been using when they wanted to apply the notion of information to biological data and biological phenomena. So I'm specifically going to relate to a information theory, which is a technical term for Shannon's information theory or communication theory, where Shannon did lots of quantification studies about information, about how to transfer information. So let's just start off with a quote by Shannon about the concept of information. So as you all know, Shannon is the father of information theory. He started off in the mid-40s really working on it, came out with a paper in 1948. And he said it is hardly to be expected that a single concept of information would satisfactorily account for the numerous possible applications of this general field. So you already, he used the concept of information, but he provided this disclaimer that he knew already that there are going to be a lot of people that will maybe not like him using that sense of information, because information means many things to many people. So back in 2000, John Maynard Smith, who's the famous British biologist, came out with a kind of a more philosophical paper about the concept of information in biology. And what he really said, in essence, is that people are looking at the DNA. After the DNA revolution, people have become more interested in how to apply the notion of information in biology, because DNA has lots of properties that are very well conceptualized within an information or an informational framework. So for instance, it can be looked as a carrier of information from one generation to the next. The code itself is abstracted from the substrate. It doesn't really matter if it's nucleotides, how the nucleotides are made up, or it could have been proteins, or it could have been something else. So there's a level of abstraction. And so there's also information transfer, which there's this, I think he referred to the central dogma, so he really saw information flowing from one point to the other in a unidirectional manner, which is pretty much what most people see in heredity. And he also talked about the colloquial use of informational terms like redundancy, translation, transcription in biology, which are all really, they come from his information theory, or, I'm sorry, from Shannon's information theory. But he kind of concluded that the actual theory of Shannon cannot be really applicable to this type of talk in biology, because it really is a syntactic theory and not a true semantic theory. And looking at all these properties of DNA, we cannot really use Shannon's theory to come up with something interesting. That was his conclusion. So generally, philosophers since and even before tended to agree with Maynard Smith that most of the interesting senses of biological information, namely mostly semantic and functional, cannot be captured by information-theoretic concepts in the technical sense. And actually, Shannon himself was skeptic about attempts to expand his own theory into other fields. He said that up front. So let's look at some exceptions that have been that have came out since by some people. For instance, Dawkins wrote that natural selection reduces the prior uncertainty, really in the genetic sense, in genotypes. So he says, in Shannon's sense, contributes information to the gene pool of the next generation. Like a certain attempt of looking at Shannon in the context of DNA, really. There were some other attempts, for instance, a lot of them focused on a notion that, or on the understanding, their understanding that really DNA captures information, extracts information from the environment through selection, through the process of natural selection. So it was kind of a claim, but not really demonstrated, I would say, not really convincingly, but it's still a claim that comes on and off. We can see that in a few papers. So for instance, I'll take the middle one there. The complexity of an organism can be estimated by the amount of information its genome encodes about the environment within which it thrives or is adapted to. Okay, or selection causes population to accumulate information about the environment. So this is the kind of the type of information which these authors later use principles and concepts from Shannon's theory, like mutual information, like actually measuring information with relation to genetic data. But so these are a few attempts, really looking at how selection works and what selection captures in terms of information. But none of them really came with a solution to quantifying the amount of information in a single DNA strand or in a single genome. For instance, a famous paper by Adami from 2000 said that it is impossible to determine which and how many of the bits of the string, really the genotype, correspond to information about the environment. It's still the same theme about selection extracting information from the environment into genotypes. But it's really impossible to look at a single strand, for instance, like the one painted up there, and saying whether how much information is inside there. You'd have to have some sort of a distribution, like if you were a population view. So taking a set of genotypes and looking at the frequencies of the various alleles, and then you can use Shannon's theory, which relates to frequencies of messages. So this is one expression they came up with for information in relation to sets of genotype. I'm not going to go into that technically, but this is another attempt. So another interesting idea was that there's a sort of a dependency between the actual environment, or coded somehow, and cues that it provides to organisms which undergo adaptation. And so they were looking at the mutual information, which is a technical term which we'll look at from information theory, between the cue and the environment. That's another attempt. So let's go back or forward to Shannon's communication theory. So this was the paper that came out in 48, that he came out with in 48, a mathematical theory of communication. Notice that he termed it theory of communication, not theory of information, but these days it's more known as the information theory. And there's a nice story where he writes somewhere, von Neumann told me you should call it entropy, his measure that he came up with. You should call it entropy for two reasons. In the first place, your uncertainty function has been used in statistical mechanics under that name, so it already has a name. In the second place, and more important, nobody knows what entropy really is, so in a debate you will always have the advantage. So I took that seriously, and I also call my measure, I'd like to call it something like entropy, so I can have an advantage in any debate. So what is Shannon's entropy, really? We're not going to get into entropy in the statistical mechanics sense, but really what's important to understand, and this is kind of tricky even for people that write about Shannon's sense of information, is that information for Shannon is the uncertainty that is removed from an initial state of uncertainty when you receive a message. Say you receive a message through a noiseless channel. He then looked upon the situation where the channel has noise, but just in general, there is some uncertainty before you receive a message. You don't know what you're going to receive. The reduction in uncertainty is the information. As the more there is a reduction of uncertainty from the initial state, the more information you have received. This is the information sense for Shannon, which has been used ever since in communication. And another thing that Shannon said, which should interest us, is that the messages may have a meaning, but the semantic aspects of the communication that he deals with are irrelevant to the engineering problem, and he is interested in the engineering problem. Okay, so let's say a few words about his measure of information and his conception of information. So it's really concerned with the probabilities of the messages, not with the actual messages themselves. So he's only and his measure of information is a measure of the probabilities of the occurrence of messages or of the distribution of messages. So it's only a function of the PI are the probabilities of the various messages. It's really mostly an average because he wants to take it all in and pack it into a single measure. So it's an average over the distribution of these of the messages over all the PIs, and it doesn't really ever look at the information content of a single message, but rather just on the possibilities of all the messages. Apart from you can look at what's called surprisal, which is just a single message, but it's not really used. Another sense of information out there is Kolmogorov Complexity, which is a different sense of information. It really measures randomness. It's not really a measure of information, it's more a measure of randomness. And then it doesn't look at probabilities, it looks at the actual message, and it looks at the single message. So for instance, if you have two messages here, first one A, B, A, B, A, B, A, B, intuitively you would understand that that contains less information than the message below it, which may be random or maybe not in a sense, but the first one at least can be described by a very small compressed string, thus it has less information. That's Kolmogorov complexity. There's another sense of information out there that's called Fisher information. Fisher was a biologist and a statistician, which is another measure. It's kind of like Shannon information, Shannon's entropy, which we'll look at, but it's more focused on giving us the information that a distribution has about a particular parameter of the distributions, like the mean or the variance. Okay, what do I want to do? I want to measure the information about what's called in biology population structure, or more in population genetics, specifically population structure. Population structure is really the substructure of the population. That is, if you sample a large group of people or other organisms or whatever across a large range, there is a substructure. They are composed of subpopulations with different gene frequencies in each one of them. So there is a possibility of extracting that structure or that substructure, classifying unknown individuals where you get a genome, you don't know where it's from, you want to say that with most probability it belongs to population A rather than population B. Things like that. A lot of work on classification and clustering is now being done on empirical basis. So I want to quantify the information that is available in genetic data for doing these sorts of things. So there are really three things that I can do. I can give the amount of information which is really the potential for classifying unknown genotypes to their source population, like I explained before. Another thing that we can do is to somehow describe the clusteredness of genetic data, to describe how much they are really clustered, because they can be very homogeneous, the data, sample from different populations, if the populations are very close or similar. Or they can be very clustered and distant. This is a different sense of information. It's kind of hard now in a single talk to explain why it's different, but it's very similar. If you have more information in the first version, you'd have more information for a type of quantification of the second version, but they're not the same. People that are more versed in statistics, I'll say that the first one is a supervised learning problem. The second one is related to an unsupervised learning problem in statistics. And a third sense is to look at the information in a single genotype. And we've seen a few slides before that Adami from 2000 tried to look at that and said it's just impossible to say how much information there is in a single genotype as it relates to his sense of information, which is information that is extracted by selection from the environment into the genome. Because you don't know how much, you have to have a collection of genome and look at the frequencies and then get a sense. So these are the three steps. So I'm going to talk about A, because right now I'm actually working on B and C, and we don't have really much time to talk about them. I'm going to talk about the first sense. So what is classification or supervised learning? Classification is a very simple notion. If you have data coming from different groups or populations, say you have 3 sets, 1, 2, and 3, and you know that they're coming from these sets. You know you have 3 samples. You can build a classifier. A simple classifier would be just a straight line over there, like these 2 straight lines. And then if you have an unknown genotype, you don't know its color, but it lies here, then you know that it belongs to 2. If it lies here, it belongs to 1. If it lies here, it belongs to 3. And so that's how you classify. So this is a classification problem. Same thing on the left there, which is a more complex classifier. It's not a straight line. Okay, and I'm going to borrow from Shannon. So what I do really is use a lot of principles from Shannon to arrive at a measure of information, like Shannon did arrive at his measure of information. So Shannon used what's called an axiomatic approach to define a measure. That means that he thought of, he imagined, dreamt of, or whatever. Just intuitively, you come up with a few criteria or a few properties which any information measure should comply with. But these properties are like axioms, so it doesn't matter how you come up with them, as long as they make sense to you and to maybe others reading a paper, then they're okay. It's an axiomatic approach. And another thing that I borrow from Shannon is a conceptual link between information and uncertainty, between these two concepts, information and uncertainty. Because for Shannon, remember, information is a reduction of uncertainty at the receiver side once he gets a message from the sender. For me, you'll understand that my information about population structure is really a reduction of uncertainty about where an unknown individual, an unknown genotype, should be classified. And thirdly, I also measure information as an average on all the possible outcomes. That is, not just one genotype where I can classify it, but if I take all the unknown genotypes that I can ever get and average information across all of them, this is the measure of information on that hyperpopulation, let us say. Okay, this is from Shannon's paper. So he came up with, he dreamt of, I always like to think of it that he like dreamt of them because he doesn't really have to justify how he got to them. As long as they sound intuitive and people agree with that. So let's just go over them. There are only 3, so they're very basic and very intuitive. You'd see, so the measure, his measure is H. It's over a set of probabilities. of messages that can be received, okay? P1 to Pn. So H should be continuous in the Pi, right? So if there's a change in the probability of one of them on account of a change in another, H shouldn't be jumping around like crazy. It should be continuous in the Pi. If all the Pi are equal, right? So if say you have two options and you have 50% for each option, like you're throwing a coin, that's the maximal amount of information. So in this sense, it should be monotonic increasing as a function of n. So if I have two possibilities, that's two equal possibilities of a message, that's like a certain amount of information. But if I have three equal possibilities for messages, that's more information, and 4 is more information. That also makes sense. Once you have more possibilities of messages, equal probabilities, probably you would have more uncertainty there, right? And 3 is a bit more tricky, but it's also, when you think about it, it's very intuitive. It just means that it's decomposing the branching of options. You see here, if you have 3 options, half, third, and a sixth, where you can go to, these are the probabilities, right? They add up to 1. It's the same thing as starting from half, half, half to 1, and then half to the other, and then 2/3 and a third. It's the same, it's just a different path, but you get to the same 3 probabilities. So that's decomposition. So with these, he came up with the famous, really Boltzmann's equation in statistical mechanics. Turned up to be the same, just looked the same. Until now, people are arguing whether there is really something meaningful in that it's the same exact expression as in entropy in physics. But he came up with a thing, and he proved that it's unique and that it satisfies the 3 options. And notes that it's up to a k, that it doesn't really matter the amount, because it's just relative to other probability distributions. You can say there's more information or less information, but it's up to a k for a certain, for any distribution. So there were other approaches in the literature. It's not just that Shannon, some approaches with relation to biology, with things that really are close to what I'm doing. Already in 1917, Richard Lewontin, a famous biologist from Harvard, used this type of axiomatic approach to define a measure of differentiation between populations, which is kind of close to my measure of information, but it's not quite, because it's really a much more simplified view of population structure. It just looks at, say, the distance between the averages of 2 populations, but not really on the whole structure, which we'll see. But he started off by just mentioning that because he started off also with an axiomatic approach. He defined a few characteristics that should be complied with and found a measure. It actually found a measure based on Shannon's. Similarly, it's done for complexity measures, which is a big topic I'm not going to get into now, but it's harder than information measures because no one really knows what complexity is. People can kind of understand what information is. So what is my data? My data is really genotypes. So you take a genotype, there are lots of sequencing activities going on in the world all over labs. You take a genotype, you sequence, for instance, SNPs. If you know what SNPs, single nucleotide polymorphisms, are the smallest changes in the genome, just single nucleotides that vary among populations, within populations and among populations. So each one is a letter, really, G, T, C, or A, right? So this is a genotype. This is just a string, right, of places that you look at which vary within a population. So this is the data. You have many strings like that. This is your data of populations, and you know where each string comes from, because I'm starting the supervised problem. I know, so I'm getting, say I have 2 populations, I'm getting a bunch of strings like that for one population, a bunch of strings like that from another population. Then I use that data to generate some sort of measure of how unknown individuals, unknown strings, the strings that we know the string, but we don't know where they came from, the amount of information that is in the whole data that will allow an unknown genome to be classified correctly. So we can look at it. Yeah, that's why it looks different than in my PC. But this is really, what should be here is a 3-dimensional cube because what we have here is two populations, the green and the blue, and 3 genes for each genotype. And we have here a sample of genotypes. So for instance, on this corner here, on this vertex, you'd have all the genotypes that are, for instance, CTT. Okay, they have 3 nucleotides, these 3 nucleotides. So there are 8 possibilities if you have 3 SNPs, right? 3 genes, 3 small genes, which are called SNPs. There are 8 possibilities so there are 8. So it's a hypercube, 3-dimensional hypercube. So if I'm taking samples from a green population and from a blue population, you already see here, this is really simple, just it's really simple genotypes and just 3 SNPs. You already see here that there's this kind of a separation between the populations, right? The green one is more up there. I hope you can't really see that it's a cube. And the blue one is more down here. They're kind of separated, but they're not really separated. So if I just get someone here on this corner, let's say someone on this corner, it's not really clear if I should classify it to the blue or the green, but someone on this corner is probably more green because I got more samples of green there. So that's my problem. I want to see how much information there is in a structure of genotypes. So mathematically, I'm getting a list of frequencies of genes in one population and a list of frequencies of genes in the other population. So that's P is the list in one population, P1 to Pn, and large Q is the list of frequencies of genes in the other population. So that's what I have to work with. And now I have to generate the amount of information that is there to classify. So with diploid populations, so that was haploid populations, a really simple model. Diploid populations is really like humans are diploid. They have 2 sets of chromosomes. So they don't have, and even if we're looking at SNPs, you can have, it's usually denoted small a and large a. For instance, if the 2 genes on the homologous genes are not the same on the same locus, or you can have small a and small a if they're the same, larger or large a. So there are actually 4 possibilities, but really 3 in most cases. So the hypercube looks more complicated. It's still 3 genes. You can see here again, the blue samples are more there, the green samples are more there. The small dots are the centers of the populations. You can see that there is a substructure. There's a substructure there, and I want to quantify it. Okay, so similar to Shannon, I'm going to dream up a few axioms that my information measure, or any information measure that does what I want, should comply with. Turns out I couldn't dream of 3. I had to come up with, you'll see, a much larger number. We'll go over them briefly, and just to see the meaning of things. So for instance, 0, this, like Shannon also has a 0. So it means there's 0 information, principally if all the frequencies in that I got from population 1 are similar to the genes, the frequencies of genes in population 2. Really, the populations are not really differentiated. We sampled here, they're the same frequencies. Makes sense that my information, any information measure should be 0. Shannon has something like that as well. Non-negativity, of course, there's nothing like, it doesn't make sense to talk about negative information. It has to be greater or equal to 0. Again, Shannon has something like that as well. Bound. Unlike Shannon's information, mine has a bound. I want it to have a bound. I don't want it to go to very high numbers depending on the distribution. I always want it to be bounded by 1 because 1 means something to me. 1 means, remember, it's really a measure of uncertainty. 1 means that there is no uncertainty. That I am certain that I know how to classify unknown individuals. So 1 is really means that, so I want it to go from 0 to 1. Now this is really important. This is a reflection of lots of studies that have been going on, empirical studies, and some theoretical insights from other people as well. That I want a criterion that's called performance. What's that? I want it to increase. Shannon also has something like that. Remember that it increases as you have more possibilities. I want mine, I call it CN, by the way, instead of H. So C with the subscript n, meaning the number of genes that I take. So n would be 3 in the case of that hypercube. But generally, in real terms, it's usually tens of thousands, or it can be hundreds of thousands of SNPs. So I wanted to increase generally when I have more genes to sequence, when I get more genes to sequence, or when I look inside and I sequence more SNPs or genes or whatever, or other types of polymorphisms. It makes sense that I will have more information about the substructure. So that's performance. And here you have something to give you a sense of it. Say these are the two populations, and they're like two spheres. So if you, in a single dimension is like looking at one gene. So you have this option here. You can see that the overlap, that's the area of uncertainty, is a third of the length of the total. The overlap of these two. When we expand to 2 dimensions, like we're adding another gene, we're looking at 2 genes now, the overlap is about, I'd say, a fourth of the total area. But when you're looking at spheres in 3 dimensions, it's already like the overlap, you can't see it too good here, but it's kind of like a fifth of the total volume. So really, it's a well-known phenomenon in statistics, really, that as you increase the dimensions, I can't decrease it to 4, I wish I could. Fortunately, we're bound now by 3 dimensions, visually, conceptually as well, mostly. But as we go to higher and higher dimensions, the groups become more separated and the overlap decreases. So that's kind of the intuitive justification for my performance criterion. Another one is convergence. Remember, it's bounded by 1, but so I don't want it to go, as I add more genes, as I sequence more genes, I don't want it to go up to infinity. It should asymptotically rise something like this. I would want it to be something like this, right? My informativeness or my information measure. And I'm also basing it on empirical results. For instance, this thing on the left, this graph on the left, which is from an empirical study about how the misclassification rate, which resembles what I do, the error rate of classifiers goes almost to 0 as you increase the number of loci. Loci is the places in the genome, or the number, you can look at it as the number of genes or the number of SNPs. Okay, another one that I have to have. Okay, these are like simpler ones. Neutrality, I'm not going to go over them, spend a lot of time on them, but it just means that when I add, if I look at another location in the genome, but it's not really informative because I have the same frequencies of genes in that locus in both populations, I shouldn't be adding more information. And continuity, remember Shannon's continuity, that if you change slightly the probabilities of messages, then H should not jump. Similarly for my CN. Another one is dominance. Briefly, I'll tell you what that means. That simply means that if at a certain location in the genome, there is a complete differentiation between the populations. That is, all the people from one population have gene A at that location, and all the people in the other population that I sampled have gene B. So they're fixed, but fixed to different genes in both populations. Then intuitively, it would be very easy to classify an unknown genotype, right? I just look at that place. I don't have to consider all the genes, and I know how to classify. So I would want my CN to be 1, or to asymptotically reach 1 as the differences of frequencies go to 100% difference. Or in terms of probabilities, they go to 1. So you can see it here in the graphs, right? If all the greens are here, they are distributed, but only along 1 dimension. And all the blues are up there, then it's very easy to get one. Because if you get it here or here, you know that it's green. But on the left, you have something without dominance. So information is not 1. Okay, I'm not going to get into these. These are more intricate things, but they are so, I'm pretty much convinced that they are required. So they are more intricate. Another important one, we're almost reaching the end. I'm already at 13. Remember, Shannon had only 3 he could work with. I'm like I envy his, but I can't go with 3 because there are some others that are really important. And for Shannon, he really proved that with 3, he uniquely mathematically proved he can come up with his measure, famous measure. So this one means that, remember, I'm not just getting samples from 2 populations or 3 populations. I'm just using 2 as a simplified model, but my model should work with any number of populations that we're starting off with, source populations. But I'm also getting the relevant sizes of them. So if one is very small, one is a small population from Papua New Guinea, one is a large population from China, okay, and I'm getting, so I know that if I get an unknown individual, there's prior probability without even looking at the data that he's going to come from China, just by Bayesian logic, right? So that prior, I call it alpha. It's between 0 and 1. So 0 means that like there's 0 prior probability that it comes from population A, and that goes from 0 to 1. So my measure should include that, and includes that. But this criterion means that if the measure is the prior is at the extreme, then I have 100% information. I don't even need to look at the data itself. If one population is of size 0, for instance, then of course an unknown individual, the probability that it will come from there is really low. Okay, and the last one is subadditivity. Subadditivity is something interesting. Everyone knows a bit of geometry and math. There's something called subadditivity or the triangle inequality. It just means that if I look at my measure as a distance measure, so if I have 3 populations, not 2, I want it to satisfy the triangle inequality. So the distances between 2 populations plus the distance between that population and a third would always be greater than the distance between the first and the third population. Why? Because then I can talk about it as a metric also, as a metric. And metrics have nice qualities, and people like to talk about metric. They can talk about it as a distance. It's not only an information measure, it's also a distance measure. And all the other measures of differentiation, like Lewontin's that I considered, and all the ones FST that are used right now are not really metrics. So who knows? Maybe in the future it will have an advantage, but I wanted it to be a metric. So I wanted to comply with the triangle inequality. So just to sum up, a metric has to just support 4 simple criteria, and these are 4 of my 14 properties. So I'm not going to go over these, but these are a posteriori properties. A posteriori properties meaning that I don't really want to specify. They're not intuitively understood. I can't intuitively justify them as properties of my information measure. But I found out later that they are properties. Now, Shannon had exactly the same kind of strategy. He wanted to find the minimal set that are axioms, term them as axioms, and then after he found his measure, the famous expression, he found that it complies with many other properties, but he didn't need, and he couldn't justify having them as axioms. So similarly, I have a few, 3, that I can't justify them as required, but they are interesting nevertheless because they come out after I found my measure, but it's still listed here. Some conceptual differences between my measure and Shannon's, again, to stress. So H is limitless while mine is bounded. H, which is Shannon's entropy, is measured by bits, right? We haven't discussed that really, but really measured by bits. It's like, so everyone now talks about bits and bytes in this computer, but really it's an invention, probably, it's kind of an invention by Hartley just a few years or more than a decade before Shannon, but still there is a sense that we can measure the information in a communication channel in bits to whatever base, usually base 2, so logarithmic base 2, as we know it now. Well, but mine doesn't have, there's no sense that mine can be measured in bits because it's really a measure of, it's more closer to a measure of real uncertainty. Uncertainty of how we can know where to classify unknown individuals. So it's unitless. And H is a simple description of uncertainty. It's just we haven't really looked at the expression of Shannon, but you guys can look later, but it's really just a measure of the uncertainty in a distribution. Mine should be really a procedure-related, just a conceptual thing. It should be a procedure-related measure of uncertainty because it really wants to measure what a certain procedure, like a classifier on genetic data, can do. And just a final note there that CN is more similar, my measure, to what's called Fisher information that I mentioned before, because it's Fisher information is a measure of the distribution, but not the uncertainty, but really uncertainty on a certain parameter of the distribution. And it's unitless as well, so it kind of more resembles Fisher information. So what I did, just to go briefly on the attempts, tried to come up with a measure. So I looked at various measures used in mathematical and statistical literature on differences between distributions and things like that, divergences. So something that comes up is a Mahalanobis distance is something very simple conceptually. Remember, this is my data and just for 3 genes. So just the distance between the 2 centers, the 2 small spheres. It's just the distance. So if you try that out with my parameters of SNPs, the parameters that I use in the context of genetic data, it turns out that it doesn't comply with several criteria. One of them is remember the priors one, which means that if the priors are really divergent, 0 and 1, then I have 100% information. You can see here that it doesn't go to 1. When the prior goes from 0 to 1, Cn is not at the top, it's just in the middle there. So for instance, it doesn't comply with that. Let's use actual information-theoretic measures from Shannon's theory. So to go over that without complicating things too much, I'm looking at the problem in terms of a random variable now, like Shannon. So each y1 to yn is really a multivariate random variable that describes the genotype, and x is the variable that describes the source population. And I'm taking the mutual information, something I haven't invented. People are doing that for various other purposes. Mutual information between these 2 variables. So mutual information is a term of Shannon, is a basic term of information theory. And I express it in terms of my parameters. So I get an expression. The same thing you can do with what's called relative entropy. Relative entropy is the difference really between 2 distributions, but in terms of entropy. You get the same expression when you use it for genetic data. That's what I found. And again, it doesn't comply with about 3 or 4 criteria of mine. For instance, the priors, again, it doesn't comply with it. It actually goes the other way around that it should, because I want my priors to go up to 1. Sorry, I want my measure to go up to 1 at the edges when the prior is 0, 1. It actually goes down to 0. So a basic information measure, 2 basic information measure concepts do not work with this type of problem. So I decided to take what's called a decision-theoretic approach. A decision-theoretic approach means that I want to use things from decision theory, not really information theory. So I actually do a really, it's not a surprising step because let's go back. What I wanted originally was a measure of information that classifiers about the potential for classifiers to succeed of classifying unknown individuals. So why not use classifiers to generate my information measure? But what I can use really is the error rate of classifiers. Classifiers have error rates, but usually classifiers unsupervised and supervised are really procedures, they're algorithms, they're not mathematical formulas, but I needed a mathematical formula because I want a measure. I want an expression for a measure. So I'm looking at the error rate of classifiers and I'm basing my information measure on the error rate of a classifier. But then I need to really I need to find out which classifiers to use and how to find their error rates. So when I do that, and I take the average, the error rate is the average, and it's really the complement of the error rate, right? If the error rate is large, then there's not a lot of information. If the error rate is very small, so you'd understand it has to be something like that. It's kind of like a complement. Then it satisfies, surprise, surprise, all the 14 criteria. For instance, it satisfies the convergence property on the left. Remember that it should go to, this is actually a printout of the measure for certain data, but they all look like this, that with more loci, right? More genes, it's going to go up and going to go to 1. And for instance, this is the dominance. Remember the dominance property, that if at a single locus there is differentiation, it's enough to look at that locus, at that gene to classify. So when the little frequencies differentiate and go to 1 in that locus, my measure goes to 1 with different alphas, which is the priors. It doesn't matter. It's just two properties that I show here. But different than Shannon's, it's not a unique measure, because I didn't prove it mathematically from the criteria like Shannon did. I dreamt of a measure, and then I tested it mathematically with all the 14 criteria. So it turns out that I can use a completely different classifier. I'm sorry, I didn't mention, I used the error rate of what's called a Bayes classifier. It's really the strongest classifier that is known, Bayesian classifier, when you have the parameters of the distribution, that is really large samples. But you don't really always have large samples, because you're sampling like 100 people there, 50 people there, or 30. You have really samples of frequency, estimators of frequencies. But anyway, so there are other classifiers that you can use if you use what's called a distance classifier based on distance. So I'm looking at the distance between the unknown individual that I want to classify and the means of each population, and I'm just classifying according to the smaller distance. But it's normalized over the shape of the distributions. So there's something called a nearest centroid classifier. If I'm using that, it also complies with all criteria. One thing that is not mentioned here is that not every classifier works. So that's kind of also an interesting result. There has to be some strength to the classifier so that it can operate as an information measure. Because I tried very simple classifiers, simpler than these two, and they fail on certain criteria. So, and we cannot know that in advance when they fail and when they succeed. We just have to test it. So I tested two and generated the mathematical expression from these two, and they work. So let's look at something that's called sampling effect. So I mentioned this briefly. So my measure was defined in terms of the PI and the QI, right? The probabilities of the genes. But really what practitioners have in the lab, again, they have finite samples from populations. So there is really some sort of uncertainty about the underlying gene frequencies when you take a small sample. If you take just 5 people of a population of millions, and you try to estimate the gene frequencies across all the loci just from 5 people, you're going to get lots of approximations, errors. So in a sense, you have less information as the sample size decreases, right? That's also intuitive, and it's a well-known result from other problems where you have sampling effects. So I want to quantify that. I actually want to have my measure compliant with the phenomenon of noise. So it's analogous to Shannon's noise in a communication channel. So it's a well-known result that classification performance is degraded with smaller population samples. I want to take that known phenomenon and have my measure comply with that. So I need to extend my measure to take into account sample sizes. So my measure will look like something like C, N, M. So N is the number of genes that I have, and M is the number of samples that I have. So if you look at the, here I did something analogous to what Shannon did. The upper diagram is from Shannon's paper, that famous paper where he, at the beginning of the paper, he really draws out his model of the communication channel. Have an information source, transmitter, then the signal goes through a channel, but there's noise introduced, just electrical noise, degrades the channel, degrades the signal, actually. Comes to a receiver, the receiver decodes the message and ends at the destination. So I can look at my kind of process similarly. I have population data, I parameterize it, so I find there are low frequencies in all the, and then there are sampling effects that I discussed, because I'm taking small samples. That's kind of like the noise that is introduced. And then at the other end of the receiver, there's a algorithm that does classification or clustering or whatever. In my sense that I talked about now, it's classification. And then we get an answer. So sampling effects in that sense are kind of analogous to noise. So I needed extra criteria. So just to make a long story short, the criteria are they're very intuitive. They just mean that a measure that takes less than an infinite sample, like takes a finite sample, has to give less information than measure that takes an infinite sample, or that knows all the parameters of the distribution. That's sampling. I call that sampling effect. The other one is sampling convergence. As the sample size increases, I want it to reflect more information that I have. So I wanted to look something like this. For instance, this is this just means that with sampling you have the continuous line, but it's always the amount of information is below that dotted line, which is for infinite sample, but it's still increasing. And here you have a case where, similarly, it increases with sample size. Just like it increases with the number of loci. The amount of information increases with sample size. Okay, so I'm actually finishing. So, okay, so I mentioned that not every classifier works. And there's really no heuristic to know which classifier can work in that sense. As to work, I mean in usable to generate a measure of information. So, last slide. Questions to you. Really, is this a convincing measure of some sense of biological information? And is it a new sense of information? And does it also suffer from not having a semantic kind of dimension?